Okay, let's unpack this. We are diving deep into the cell's inner workings today.
Hmm.
Specifically, how those instructions locked away in your DNA actually get used, get brought to life.
Right, the core engine of biology. It's what we call the central dogma,
Yeah, that famous flow. DNA makes RNA.
which then makes the polypeptide, the
Oh,
protein, the final product.
exactly. And our mission, really, for this deep dive is to simplify that first big step, transcription.
Yeah, that journey from the DNA blueprint to the, let's say, working copy of the RNA.
But also, why does this look so different in complex cells, like ours, compared to simple bacteria? It's way more complicated.
It really is. And that difference is key.
Which brings us to the big question for you listening. Why RNA? Why use this intermediate molecule? It seems, well, kind of fragile next to DNA.
But that apparent fragility, that
Yeah.
instability, that's actually fundamental to how life controls itself. It's not a bug, it's a feature.
Okay, so let's zoom in on the molecules themselves. DNA, the master blueprint, safe in the vault. RNA, the working copy out on the factory floor. They seem similar.
They do look alike at first glance, yeah. But the differences are critical for how they function.
Right. So what are the key distinctions?
Well, three main things. First, RNA is usually just a single strand, not that stable double helix-like DNA.
Okay.
Second, it uses a different base, uracil, or U, instead of thymine, T, which you find in DNA.
Uracil instead of diamine. Got it.
But maybe the most crucial difference, the one with the biggest consequences, is the sugar in the backbone.
Ah, right. DNA has deoxyribose. RNA has a ribose.
Exactly. And that ribose means it has this tiny extra bit, an oxygen and hydrogen, an OH group sticking off the number 2 carbon of the trigger ring.
Just one little hydroxyl group. Seems small.
Small, but it changes everything. That 2' OH group makes RNA chemically reactive.
Hmm.
It can actually attack the phosphate backbone itself.
Attack itself, so it can break down on its own.
Precisely. It makes the phosphodiester bonds much less stable than in DNA. RNA is inherently prone to degrading.
Okay, that sounds like a real problem. Why would you build a system carrying vital instructions on a molecule that's, designed to fall apart?
Well, here's where it gets really interesting. Yes, it's less stable, and maybe that's partly just how things evolved early on.
An evolutionary leftover.
Could be partly.
Yeah.
But that instability, combined with being single-stranded, gives RNA amazing flexibility. It can fold up into complex 3D shapes,
Like proteins do.
exactly like proteins. Think about tRNA or the ribosome itself. They have intricate structures. RNA can even act like an enzyme, catalyzing reactions. It's incredibly versatile.
So the cell isn't just tolerating the instability.
It's using it. The fact that RNA degrades quickly is a powerful control mechanism.
How so?
Think of it like a temporary memo. Need a protein, make lots of its RNA message. Job done. The RNA disappears quickly.
Ah, so it provides a built-in off switch, stops the cell from making stuff it doesn't need anymore.
Precisely. It prevents waste and allows rapid responses to changing conditions. That instability is crucial for regulation.
Okay, that makes a lot of sense. Regulation is key. So we understand RNA now. Let's look at transcription itself, the basic rules that apply everywhere in all life.
Right, the universal principles.
Yeah.
And they're quite straightforward on the surface.
So the enzyme, RNA polymerase, or RNAP.
Yep, RNAP reads the DNA template strand. It moves along it in a 3' to 5' direction
Okay, reads 3' to 5'.
and builds the new RNA strand in the opposite direction, 5' to 3'.
Always 5' to 3' synthesis, like DNA replication.
Exactly. And the new RNA molecule is complementary to the DNA template strand it was copied from and anti-parallel.
And the building blocks it uses are those ribonucleoside triphosphates, AUCG.
That's right, the RNTPs. They provide the bases and the energy. And the RNAP enzyme itself is, well, quite the machine.
How so?
It's highly processive.
Processive? What does that mean exactly?
It means once RNAP grabs onto the DNA, it doesn't easily let go. It just chugs along, adding hundreds, maybe thousands of RNA bases in one go before detaching.
Wow, okay. So it's fast and efficient. It doesn't keep stopping and starting.
Right. It saves a huge amount of time and energy. You get rapid production of RNA when needed.
But I remember you saying it's different from DNA polymerase. DNA polymerase is super careful. It proofreads.
Ah, yes. That's a critical difference, and it's a trade-off. Two main things distinguish RNAPs here.
Okay.
First, RNA polymerases can start synthesis de novo, from scratch,
Meaning?
meaning they don't need a primer. Unlike DNA polymerases, which need that little starting block, that existing 3' end, RNAPs can just sit down on the DNA and start building the RNA chain. Saves time.
Okay, no primer needed. What's the second difference?
They don't proofread.
Wait, seriously, they don't check their work. Isn't that risky? You're making the instructions for proteins.
It seems risky, but think back to RNA's instability.
Hmm.
A mistake in DNA. That's permanent, potentially catastrophic for the organism. So DNA polymerase is slow, careful, double-checks everything.
Right, high fidelity.
But RNA is temporary. If you make a mistake in an RNA molecule, well, that molecule probably won't last long anyway. It'll degrade.
And the cell can just make another correct copy quickly.
Exactly. Speed and efficiency are prioritized for RNA production because the cost of an error is low and temporary. It's a calculated risk that pays off in responsiveness.
Okay, got it. Speed over absolute perfection because the product isn't permanent.
Precisely. And regardless of whether it's bacteria or us, this whole process unfolds in 3 basic phases: initiation, elongation, and termination.
Starting, building, and stopping.
Yep. But how those phases work, that's where things get really different between simple and complex life.
Let's start with the simple guys first. Prokaryotes. Bacteria. What's their main strategy?
Speed, efficiency,
Yeah.
simplicity. And the key physical difference is they don't have a nucleus.
Right, no nuclear envelope separating the DNA from the rest of the cell.
And that allows something amazing called transcriptional translational coupling.
Coupling.
Yeah, as the RNA polymerase is zipping along the DNA, making the mRNA molecule,
Up.
ribosomes can actually latch onto the beginning of that very same mRNA molecule while it's still being made and start translating it into protein immediately.
Wow. So making the message and reading the message happen basically at the same time in the same place.
Exactly. It's like an incredibly efficient, continuous assembly line.
Yeah.
Maximum speed.
And they manage all this transcription with just one type of RNA polymerase, right? A big complex.
One main type, yes. The core enzyme has several parts, but the crucial bit for getting started correctly is another subunit called the sigma factor. Think of it as the guide.
The sigma factor, what does it do?
Its job is to recognize the starting line on the DNA, the promoter region. It makes sure the main RNAP enzyme binds at the right spot to start transcription.
How does it find the right spot?
It looks for specific DNA sequences, consensus sequences, within the promoter. The 2 most important are called the NACA35 sequence and the NACA10 sequence.
-10. Isn't that often called the Pribnow box or tatat?
That's the one.
Mm.
Tatat. Notice it's full of adenines and thymines.
Yeah. Why is that important?
Remember, AT base pairs only have 2 hydrogen bonds holding them together, unlike GC pairs, which have 3.
Okay, so they're weaker.
Much weaker. That AT-rich nectine sequence is inherently easier to pull apart, to melt open.
Ah, so the sigma factor guides the polymerase there, and that specific sequence makes it easy to separate the DNA strands and form the transcription bubble needed to start.
Exactly. It lowers the energy barrier to get things going. Minimal fuss.
Okay, so initiation is straightforward. What about stopping? Termination. How do bacteria know when to stop copying?
They have 2 main tricks for termination. Very clever mechanisms.
Trick 1.
Is Rho-dependent termination. It involves a helper protein called Rho.
Rho, what's its job?
Rho is a type of enzyme called a helicase, meaning it can unwind nucleic acids. It binds to a specific spot on the growing RNA molecule called the rut site, usually a C-rich area,
Okay, it binds the RNA.
and then it kind of chases after the RNA polymerase, moving along the RNA strand. When the polymerase pauses at a termination site,
Rho catches up.
Rho catches up and uses its helicase activity to literally unwind the RNA from the DNA template within the transcription bubble.
It just pries them apart.
Yep. That destabilizes the whole complex, and the polymerase, the RNA, and Rho all fall off the DNA.
Okay, that's one way. What's the other?
The other is Rho-independent termination, sometimes called intrinsic termination. This one doesn't need an extra protein factor.
So how does it work?
It relies entirely on the sequence of the RNA itself, near the end. The DNA template in this region has inverted repeats.
Meaning sequences that are complementary further down the strand.
Exactly. Followed immediately by a string of about 6 to 9 adenines.
Okay, inverted repeats, then lots of A's. What happens when that gets transcribed into RNA?
The inverted repeats in the RNA immediately fold back and base pair with each other, forming a stable structure called an RNA hairpin, stem loop.
Ah, okay. Like a knot in the RNA.
Sort of. And
Yeah.
right after that hairpin, you have the string of uracils transcribed for those adenines in the DNA.
U's, which pair with A's in the DNA template.
Right. And AU base pairs are the weakest possible connection, remember. Only 2 hydrogen bonds.
So the hairpin forms, and that somehow yanks on this weak UA region.
Pretty much. The formation of the hairpin structure physically disrupts the polymerase complex, putting strain on that weak RNA-DNA hybrid region made of U's and A's. It's enough to destabilize everything.
And the whole thing just falls apart. The RNA is released.
It falls right off.
Huh.
Elegant, isn't it? Just uses the structure of the RNA itself.
It really is elegant. Simple, fast, efficient. But now, eukaryotes, us, everything changes, doesn't it?
Everything. Simplicity goes out the window, replaced by layers upon layers of complexity and regulation.
And the biggest physical change is that nuclear envelope, the nucleus.
That compartmentalization is the game changer. Transcription happens inside the nucleus.
But the ribosomes, where proteins are made, are out in the cytoplasm.
Right, so you can't have that coupling we saw in bacteria. The RNA message, the pre-mRNA, has to be made, then processed, then actively shipped out of the nucleus before it can even find a ribosome.
A whole new set of steps. And instead of one main RNA polymerase,
We have at least 3 major ones in eukaryotes. Pol I makes ribosomal RNA, Pol III makes transfer RNA and other small RNAs.
and the one we really care about for making proteins is RNA Pol II.
Pol II is the star for protein coding genes, yes. And getting Pol II started, initiation, is vastly more complicated than just sticking a sigma factor on.
No sigma factor. What takes its place?
A whole committee of proteins called general transcription factors, or TFs.
General TFs, TFIA, TFIB, TFID, etc.
Those are the ones. It's a stepwise assembly process. You need this whole crew to gather at the promoter before Pol II can even think about starting.
Sounds like a major production just to get going. Is there one key player that starts the assembly?
Yes. The first one on the scene is usually TFAI. It's a complex itself, and crucially, it contains the TATA binding protein, or TBP.
TBP, so it finds the TATA box in the eukaryotic promoter.
Exactly. TBP binds to the TATA box, and that binding kind of bends the DNA, creating landmark. It acts as a docking platform for all the other TFII factors, A, B, F, E, H plus RNA Pol II itself, to assemble in the right order.
Okay, so a whole pre-initiation complex has to be built.
Right. And it gets even more complex because often you also need this huge protein complex called the mediator.
The mediator, what does that mediate?
It bridges the gap, literally, between the core promoter machinery, the TFs and Pol II, and other regulatory DNA sequences that might be really far away, like enhancers or silencers.
Ah, so it connects distant control switches to the main engine?
Precisely. It integrates signals about whether the gene should be turned on strongly, weakly, or not at all, and communicates that to the polymerase. It adds another massive layer of regulatory control.
Okay, complexity at initiation is clear. What about termination for Pol II? Is that complicated too? Pol I and Pol III use methods kind of like the bacterial ones, right?
They do, yeah. Pol I termination resembles row-dependent, and Pol III resembles row-independent. But Pol II, it has its own completely unique and, frankly, kind of brutal way of stopping.
Brutal! How so? Why does it need a special method?
It ties into the processing that the RNA needs later. So transcription goes past the actual end of the protein coding sequence. Then the RNA gets cut, cleaved, at a specific site.
Okay, it's cut. So you have the useful mRNA piece.
And you also have this piece of RNA still being transcribed, dangling off the back of the polymerase, but its front end, the 5' end, is now exposed because of that cut.
An exposed 5' end. Is that bad?
It's a target for an enzyme called RAT1 in yeast, or its equivalent XRN2 in humans.
Yeah.
This is a 5' to 3' exonuclease.
Exonuclease, meaning it chews up RNA from the end.
Exactly. RAT1 jumps onto that newly exposed 5' end of the trailing RNA and starts degrading it rapidly, like a molecular Pac-Man, moving along the RNA towards the polymerase it's still transcribing.
It chases the polymerase down by eating its own tail, essentially.
That's a great way to put it. It literally chews up the RNA track it's moving on until it collides with the RNA polymerase complex.
And that collision.
Knocks the polymerase off the DNA template. Termination achieved.
Wow. That is pretty aggressive. But what about the actual mRNA molecule, the important part? Is it safe from RAT1?
Yes. Crucially, by the time RAT1 gets involved, the actual protein-coding mRNA part has already received its first protective modification, the 5' cap. RAT1 can't touch it.
Okay, that makes sense, the 5' cap. And that leads us perfectly into the final stage for eukaryotes, this essential post-transcriptional processing, molecular editing.
Right, because unlike prokaryotes, where the gene sequence usually directly matches the protein sequence collinear, eukaryotic genes are interrupted.
Interrupted by?
Non-coding sequences called introns. Interspersed between the coding sequences, the exons. The initial RNA transcript, the pre-mRNA, contains both.
So it's a rough draft. It needs editing before it can be used.
Exactly. 3 major modifications have to happen in the nucleus before it's considered mature mRNA, ready for export.
Modification number 1. You just mentioned it, the 5-foot cap.
Yep. A modified guanine nucleotide, 7-methylguanosine, gets added to the very beginning, the 5' end, but it's added backwards.
Backwards.
Yeah, through a unique 5' to 5' triphosphate linkage, not the usual 5' to 3' link found in the rest of the RNA chain.
Why the weird linkage? What's the point?
Protection. Most enzymes that degrade RNA, like RAT1, look for a standard 5' end. This weird 5-foot cap acts like a lock. They can't recognize it or bind effectively.
So it shields the front end from being chewed up.
Exactly. It also plays a key role in getting the mRNA out of the nucleus and helps the ribosome recognize where to start translation later on.
Okay, cap on the front. What's modification number 2?
That happens at the other end, the 3' end. It's the 3-foot poly-A tail.
Poly-A meaning many adenines.
Lots of them. After the pre-mRNA is cleaved near its end, remember that cleavage step before RAT1 gets involved? Another enzyme comes in called poly-A polymerase.
And it adds adenines.
Yes, anywhere from 50 to 250 adenine nucleotides are added onto that newly created 3' end. Importantly, this happens without a DNA template. The enzyme just adds A's.
It just knows to add A's. How does it know where?
The cleavage site itself is determined by a signal sequence in the RNA, typically AAU-AAA, located a bit upstream. Once cleaved, polymerase goes to work.
And what does this long tail of A's do?
Several things. It helps stabilize the mRNA molecule the longer the tail.
Yeah.
Generally, the longer the mRNA lasts in the cytoplasm. It also aids in the export from the nucleus and is important for efficient translation initiation.
Okay, cap on the front, tail on the back. What's the third and maybe most dramatic modification?
RNA splicing. This is where the non-coding introns are removed and the coding exons are precisely joined or spliced together.
Got to take out the fluff and stitch the important bits together.
Exactly. And the precision here has to be absolute. Being off by even one single nucleotide would shift the entire reading frame downstream.
Leading to a completely wrong protein sequence. Garbage, basically.
Total garbage. So, how does the cell ensure this precision? There's a splicing code within the intron sequence itself.
A code, like specific sequences.
Yes. Almost every intron has 3 conserved sequence elements that are recognized by the splicing machinery. There's the 5' splice site, almost always starting with GU.
Okay, GU at the beginning of the intron.
The 3' splice site, almost always ending with AG.
AG at the end.
And somewhere inside the intron, there's a crucial adenine residue called the branch point.
GU, AG, and a branch point A. These signals tell the machinery where to cut.
They are the key recognition sites for the machine that does the cutting and pasting. The spliceosome?
A spliceosome, what is that?
It's huge. A massive and dynamic complex made of both proteins and specialized RNA molecules.
RNA molecules doing the work.
Yes, these are small nuclear RNAs, snRNAs, known as U1, U2, U4, U5, and U6.
Yeah.
They associate with proteins to form small nuclear ribonucleoproteins, or snRNPs.
SNRPs.
NERPs, exactly. Molecular biologists love their acronyms.
So how did these SNRPs, these snRNAs, actually splice the message?
The snRNAs within the SNRPs use base pairing to recognize those conserved sequences we talked about, the 5-foot splice site, the 3' splice site, the branch point A on the pre-mRNA.
They stick to the right places using RNA-RNA interactions.
Right, and this binding brings the exons close together and loops out the intron. It causes conformational changes, bending the pre-mRNA into the right shape for catalysis.
Snip, snip.
2 key cuts happen. First, the 5' splice site is cut, and that free end of the intron is covalently linked to the branch point A, forming a characteristic loop structure.
Called a lariat.
Exactly. It looks like a cowboy's lariat. Then the 3-foot splice site is cut, releasing the lariat-shaped intron, and the 2 exons are simultaneously joined together with perfect precision.
Wow. An incredibly precise molecular surgery performed by an RNA protein machine.
It really is. And the amazing thing is, this complexity allows for incredible regulatory possibilities.
How so? Beyond just removing introns?
Yes, through alternative splicing. The cell doesn't always have to splice the same way.
Meaning it can choose to include or exclude certain exons.
Exactly. Or use different splice sites. So from a single gene, a single pre-mRNA transcript, the cell can generate multiple different mature mRNAs by mixing and matching exons.
And each different mRNA would code for a different protein.
Or related proteins with different functions. You can also have multiple sites for cleavage and polyadenylation, adding another layer of variation.
So one gene can actually produce a whole family of related but distinct proteins depending on the cell type or situation.
Precisely. Think of the calcitonin gene. In the thyroid gland, it's spliced one way to make calcitonin, a hormone regulating calcium.
Okay.
But in brain neurons, the exact same pre-mRNA is spliced differently, skipping an exon, to produce CGRP calcitonin gene-related peptide, a neurotransmitter involved in vasodilation and pain.
Wow. Same gene, totally different outcomes just based on splicing. That's huge regulatory power.
It's a major reason why complex organisms like us can have such diverse biology with a relatively similar number of genes compared to simpler organisms. It vastly expands the coding potential of the genome.
Okay, so let's pull this all together. What's the big picture for you, the listener? When you trace this journey DNA to RNA to protein, it's clear there are two fundamentally different strategies at play.
Yeah, prokaryotes go for raw speed and efficiency, coupling transcription and translation. Simple machinery. Get the job done fast.
While eukaryotes, well, we've traded that speed for control. Incredible multi-layered control.
Compartmentalization, complex machinery like pull-to-2 initiation, and the spliceosome, extensive RNA processing, it all adds checkpoints and decision points.
And that idea we started with RNA being unstable, it seemed like a weakness.
But it's actually central to that control, isn't it? Its temporary nature allows gene expression to be rapidly turned on and off,
Right.
and its ability to fold and catalyze, like in the spliceosome, makes it incredibly functional.
The instability isn't a flaw, it's the foundation of regulation. The messenger's fleeting nature is its power.
Exactly.
Which leads to a final thought, something for you to maybe chew on after this deep dive. We talked about the spliceosome, this massive machine, and how the really critical catalytic steps, the breaking and joining of RNA bonds, seem to be carried out mostly by the centRNAs themselves, not the proteins around them.
That's right. RNA catalysis is at the heart of splicing.
So, if DNA is the stable archive, the hard drive, and proteins are the workhorses, doing most of the jobs in the cell, what about RNA?
It carries the message, yes, but it can also fold into structures, and it can catalyze fundamental reactions like splicing.
So the final provocative thought is this. Given RNA's versatility carrying information, structuring itself, and performing catalysis, could it be that RNA wasn't just the messenger, but perhaps the original architect of this whole system? Maybe it's more central, more anciently powerful than we usually give it credit for.
Something to think about. Is the messenger actually running the show?